I'd like to introduce the chair of Vision Quest 2012 in Toronto and our first speaker this morning, Dr. Elise Heon. In fact, Vision Quest would not be the same without Dr. Heon. She's the Ophthalmologist-in-Chief and Director of the Ocular Genetics Program at the Hospital for Sick Children. She's an ocular geneticist and a professor of ophthalmology at the University of Toronto and holds the Mira Goddard Chair in Vision Research. She's also a longtime member. You see, busy people always are doing things for us. So Elise is also a member of the Foundation Fighting Blindness Board of Directors, and she's the principal investigator on our patient registry. So here, without further ado, and to talk about the state of Canadian vision research in Canada is our good friend, Dr. Elise Heon.
Thanks, Sharon, and dear friends, since I think I know most of you in the audience. And it's really a pleasure, again, to have the opportunity to chair Vision Quest this year of 2012. I can't believe it. I think I've been doing this for over a decade now. I've been asked to talk to you about what's new and exciting, and I decided to modify a bit what I would share with you, because there's some important facts that I would like to make sure that you know of. So I wanted to first talk about the genetics of eye disease. So how many in the audience have had, who are affected, have had their genetic defect identified? It's pretty good, huh? So we'll talk about that. And so we've known for a long time that many types of retinal dystrophies are due to changes in the genes in your body. And that we know that it's important to know about these genes because that's how we understand about the nature of disease. And that is an essential step for the development of treatments. So last year I confessed that 20 years ago we thought that there was 2 genes for RP. We were pretty excited about that, and actually someone reminded me that I had seen them at that time, and I was really excited that we knew everything about RP, and that the only thing we needed was a treatment. So here we are a little while later with over 200 genes associated with retinal degeneration, and we're just now really starting to understand why do the cells degenerate. And this has been essential to thinking about the different options in the development of novel therapies. One of these good examples is the gene replacement therapy. If we didn't know about the genetics, we couldn't even consider such an option. The big poster child of gene replacement therapy has been the Leber's congenital amaurosis, which is sort of a congenital or very early onset form of RP that's due to mutations in the RP65 gene. And I'm proud to say that many of our patients have had access to this innovation, which is a big deal. This has only been possible because of genetic studies. Knowing about the genetic defect that underlies your condition is important not only for access for gene therapy. It allows us to provide better patient management, provide you with appropriate counseling, whether family counseling or personal counseling and expectation, and also offer appropriate eligibility to certain therapeutic options. So I want you to know that FFB has been extremely supportive and very invested in genetic studies in the last 15 years. And it's allowed many Canadian researchers to make significant progress. So with the initial help of CIBC, as Sharon mentioned, FFB sponsored the development of a registry so that patients could be identified. So what the registry is, it's a tool. It's like a phone book, an anonymized phone book, where patients with a specific disease or a specific mutation can be identified and be offered the possibility of participate in certain studies. I call it a phone book provided by FFB without any name in it. It's not a research project. It's a very powerful tool. And if you wish to have your name and your eye condition entered in the registry, I urge you to look into the FFB website, as all the information is there on how to proceed. But what you really want to know then is, where's my gene? That's when people say, where's my gene? Where's my treatment? And which means, and many of you are not impressed when we talk about genetics, because it's been 20 years that you've been hearing me speak, and that your genetic defect has not been identified. So it's not that we're useless or incapable, it's just that the technology has changed a lot. And recently, specifically I would say in the last 5-6 years, opportunities have arisen that were not available 10-15 years ago. So we've been much better nowadays in identifying genetic defect. In our clinic still, just over half, maybe 60% of cases only have their mutation identified. So a big 40% of cases, we still don't know the nature of the genetic defect that caused the visual impairment. And I think that our yield is pretty good compared to other places. So why has it been so slow, and how have we been doing this? The technology that was available to us was to approach gene by gene, and it's trying to find a typo in Encyclopedia Britannica, for those of you who are old enough to remember that, and going like chapter by chapter. And so no wonder it takes over 20 years. And so, but with our educated guess, we would select certain chapters, certain genes, and luckily we have been lucky in 60% of cases, but not lucky in 40% of cases. The reason why I'm talking to you about this is that now 20 years later, the technologies change. And very soon we'll be able to sequence your entire genome for almost the same price as going gene by gene. So the cost of going gene by gene, just to give you an idea, could be up to $10,000 a patient, sometimes more, sometimes less. But we're lucky here in Ontario is that the government usually accepts our request, and it's sponsored by the Ministry of Health. With the new technology, we would be able to identify genetic defects much faster because your entire genome would be sequenced, and then we decide what we want to study. You may have heard in the press terms like whole genome sequencing, next generation sequencing, or exome sequencing. Those are all fancy terms that refer to the Ferrari of gene identification, whereas we were really on a tricycle. And so although it's great to have a Ferrari, there's some risks associated with having a Ferrari. Not that I ever contemplated that, but speed should not be the objective. Accuracy should be the objective. And I'm just going to give you some food for thought, is that once your whole genome is sequenced, sometimes you find things that you weren't looking for, that you weren't expecting to find. And that's not obvious how to deal with that. Like, I personally don't want to know if I'm going to have Alzheimer's. If I start forgetting, it's all fine. I don't want to know if I'm going to have Huntington disease. So maybe some people do, I don't. I don't have an answer on how that's going to be managed, but I want you to know that in the future, that's going to be the approach. In the very near future, that's going to be the approach that we're going to use, and we're going to have to think about those situations. But the good news is that this shifting gear technology brings us in an era of identifying genetic defect faster, which allows us to provide you more useful information faster, which inevitably allows us to put some treatment opportunities on the horizon also faster. So I think it's all good. We just need to be very aware of the different aspects of these advancements. So the next question that you're going to ask me, if I pretend we're in clinic, that's how I do this, is that, okay, so you made some point with this, now where's my treatment? Like, how come some people have their treatment and where's my treatment? And I want to make a point, like, don't be angry at me because it's not my fault. A lot of people get angry at me and it's not my fault. I try to do my best every day. And what you need to understand is that we can't really treat. If the word treat means fixing, we can't fix what we don't know. You can't really fix if you don't know why something is broken. And if you bring your car into the garage, that's what will make the difference between a good car mechanic and a bad car mechanic. Some will try many things and some will just fix the right thing. So as I mentioned, the poster child of treatments, which will not be the option for everyone, is gene replacement therapy. So gene replacement therapy is an approach that can be considered so far if you have an autosomal recessive or an X-linked disease, where the genetic defect is identified. So we can't do gene replacement therapy in an individual if the causative genetic defect has not been identified. So what we do in these cases where it's possible is that the gene, the missing gene or the dysfunctional gene, is packaged in a virus and it's injected into the eye, either in the jelly of the eye or under the retina, to infect the cells. So then the cells will carry the missing gene. So this has been used to date for RPE 65, as I mentioned. There's over 40, 50 cases around the world that have been done. And the good news is that it's safe. So the approach, the concept is safe. There's been no adverse event. And that's been a huge achievement. So now, the other thing to remember is that when you start a trial, and those of you who participate in the clinical trial breakout session will talk about that, is that the first phase is to ensure that you do no harm. So the first phase was a phase 1 to ensure that the treatment is not toxic. So the treatment was not delivered with the intention to offer maximum efficiency. It was delivered with the intention of showing that it's safe for the individual. So other phases of this trial will be undergoing soon, where maybe the dose will be modified and the approach will be modified to see if we can improve the efficacy. Even though the goal was not to assess the efficacy of the treatment, most individuals had an improvement in light sensitivity, which is quite remarkable. There's other trials that are now undergoing using similar principles, namely some related to replacing ABCA4, which is the gene associated with Stargardt's disease, Usher's 1B, another rare form TK. There's a group in London that have initiated a treatment trial for choroideremia, and we're proud to say that next year, thanks to the sponsorship of FFB, Ian McDonald in Edmonton will be leading our first Canadian gene therapy trial ever for choroideremia. So Dr. McDonald has spent his career into the better understanding of this disease. It's important to know that all these approaches are not treatments in the sense that they're being explored. At this point, none of them can be purchased, and none of them are approved by Health Canada at this point. Hopefully we'll get there sooner than later. So when you don't know the genetic defect, a lot of people think they're losing on something, but you're not necessarily losing on something. There's other approaches that can be considered. And one that's implemented already and that's high on the improvement list is what we call the rescue therapies, or the protective therapies, to protect the retina from degenerating too fast or too much. So these therapies are being developed to slow the death of the cells, to slow the death of the photoreceptors as they're sensing light. Example of this is simply taking antioxidants. Many of you are taking antioxidants, and antioxidants do protect the retina to some degree against cell damage. In France, there's a trial where they're injecting a factor that's called a cone viable factor. It's like to give steroids to the cone to beef up their energy. And it'll be interesting to see the results of this work. Nutrition is an important point, and there's going to be a session on nutrition in AMD, but it's pretty similar for retinal degeneration, where proper nutrition does affect your eyes. And so your eyes need antioxidant, they need the omega. Not everyone needs vitamin supplements, so we don't recommend that systematically people take vitamin A as a supplement. In some cases it's indicated, but in many cases it's not. There's a researcher in Ottawa, Dr. Tsilfidis, who's sponsored by FFB, who's using viruses to transfer genes that slow cell death using an animal model. So it will be very interesting as the work gets completed to see how that progresses. It's very important to understand why FFB supports numerous different research projects, is that numerous different avenues are considered. One that's being talked about is using, we call it repurposing of drugs. So there's some drugs on the market that serve for one reason and they can be used for another reason. You have to be careful in doing that too quickly, because you need to make sure, for example, that the side effects are worth it. One example is that many of you are either on Diamox or methazolamide or some eye drops for macular edema. So that's an example of repurposing of the drugs. You may have heard of valproic acid and so on. Another measure is restoring sights with retinal implants. So there's been two groups, the Americans and the Germans, who have done outstanding work in making the field of retinal prosthesis progress to a point where now this device is seeking for FDA approval. We do not have a site in Canada yet, but there's some negotiations that are ongoing to make that happen. What you need to understand is that the cost of those very innovative therapies is very high. And for an example, the cost of a retinal implant would be around $100,000. So it's not, you know, a Walmart type of purchase. It's we need to have a strategy. I won't talk about regenerative medicine because we have the world experts in the room, Dr. Van der Kooi and Vince Tropipi, who's going to have a breakout session about that. But just to say that some trials have been initiated, not to replace the photoreceptors, but to try to replace the retinal pigment epithelial cells. So the message that I would like to leave you with is that when we talk of treatments, our motto is first do no harm. And that's why sometimes it appears that we're slow, and that's why we force ourselves to go through clinical trials. When we talk about genetic testing, we also say first do no harm. And as we're in an era of identifying genetic defects more quickly, it's very important for us to make sure that the genetic defect that's identified is really the one associated with your disease. Because the worst thing that could happen is assuming that a genetic defect is associated with your disease and that it's not, because then it puts you at risk of having a wrong treatment. And those mistakes are easily done if you try to go too fast. So just so you know that the interpretation of these tests are not trivial, and you need to make sure that the right thing is done for you. We're in a very good position in Canada. We're lucky to have FFB because they've been generously sponsoring not only genetic research, but basic research and therapeutic research. And Canadian researchers are right on track with the other groups. So I hope this session has given you enough information. I know that it's a very superficial overview of everything that's going on. There's many hopes for now. Don't ask me when, because we don't say when anymore. We don't talk of timelines. All we can say is that things are going well. The opportunities are real. And the opportunities to offer change and outcomes are very real. And we just need to get there in the right way, in the right time frame. So thank you for your attention, and I'll be happy to address any question. We have 5 minutes for a question.
First of all, thanks for the wonderful work you're doing. I'm a parent of a 1-year-old child who's been recently diagnosed with LCA. And even though I'm a healthcare professional, I'm a specialist in cardiovascular and diabetes. And this is a great opportunity for me to learn. So first of all, thanks for the amazing work you do, because I live in that world. I know all that stuff. I don't have a question, but I do have something to ask you. You do amazing work to help people like me and all the people in this room and our families and friends. What is it we can do to help your research and your work? My limited time in FFP is I found out that, I may be wrong, don't quote me here, but I believe we don't get much government funding at FFP, or no government funding at this point. And I've been, call it shocked if I want to put it very politely, that there is no support on that front. So I'm interested, maybe if you could tell us what we could do, as simple as everybody in this room could go back and send a letter to a MPP just advising them about what FFP is, about the work we do. So just a little bit on that I'm hoping will be helpful for everybody here.
So I appreciate your question. So the question is, how can you make a difference? And so I assume you mean, how can you make things go faster? There's two important aspects to my answer. One is to raise awareness by talking about it, talking to your MPP, raising awareness about loss of vision, identifying the burden of vision loss, which is not talked about enough, so that funds can be freed towards those endeavors, because it's true that there's not a lot of government funding towards that. The other one is give generously. And I'm not saying that as a pitch, is that in healthcare, as you know, I'm sure, the costs are increasing very fast. And I can tell you, for us at SickKids, the majority of the equipment we have has been obtained through donations. If we were relying only on hospital support, we'd still be in the boonies. And so it's really by engaging the community and having the community feel responsible for the outcome that a difference is going to happen. So those clinical trials are very expensive. The clinical trial for RP65, Jerry Chader's in the room, probably cost over $20 million if you add up the years. So hopefully not every trial is going to cost $20 million, but it's a lot of money. It's expensive to be careful. And it's our culture to be accountable to society, and therefore you have to be careful. And that cannot be done without financial resources. And philanthropy will play a critical role in determining the future of where we stand.
Hello, my name is Sina. First of all, I appreciate for all things that you've done up to now. And my question is how I can get information regarding the test that they got from me for genetic. It was about maybe 3 or 4 years ago, but after that I gave my blood test, I didn't get any information back.
So the question is, how come I didn't get any information back from my blood test? So if I can only address if it's been done through the Hospital for Sick Children, often when there's no results back, it's because we don't have results back. But I would encourage you to recontact the genetic counselor who could put your file back on track. And for the other centers, if you've been seen either at North York or Mount Sinai, I'd probably recommend the same process.
Hi, I just have a question. I'm new to the FFB, and how do people go about getting the genetic testing or the genome sequencing or whatever done for retinitis pigmentosa specifically?
So genetic testing is offered through healthcare centers, and so there's different groups that have an interest in inherited eye disease. So we do at the Hospital for Sick Children. There's Dr. Nupra Bakshi at Mount Sinai. There's Dr. Radha Kohli at Sunnybrook. Michael Brands at the Toronto Western, but I understand that he refers most of his case to Dr. Nupra Bakshi. We have worked with the genetic counselors also at North York General. And so Megan Day, Megan, can you stand up? She'll kill me for this. Megan is our genetic counselor. If you have any questions, you go ask her. She's a wonderful person. Otherwise, I wouldn't do that. Thank you very much. Thank you.